This video we'll be taking a look at cardiac cell action potentials. There are two sets of cells within cardiac muscle tissue: the non-contractile cardiac muscle tissue, which makes up the intrinsic conduction system, that has the characteristics here of autorhythmicity or automaticity, that they have the ability to depolarize on their own without a signal from the nervous system, and then the contractile muscle cells themselves. Because of the intercalated discs and the gap junctions, the heart muscle tissue contracts as an organ instead of like a motor unit that we see in skeletal muscle tissue. So either all of the fibers of the heart are going to contract as a unit, so the atria will contract simultaneously and then the ventricles will contract simultaneously, or the heart's not going to contract at all. If we do have individual portions of the heart contracting at their own rates, then we have fibrillation, and fibrillation will end up not pumping blood through your body and you'll die. So we do not want independent areas of the heart, we call them ectopic foci, acting as the SA node and causing contractions or depolarizations to happen in various parts of the heart. Now, one thing about the action potential that we see in cardiac muscle tissue is that it has a much longer absolute refractory period than that of skeletal muscle. It has a period of about 250 milliseconds, and the reason why is that we need to prevent tetanic contractions. We do not want multiple motor unit summation taking place. We don't want wave summation or temporal summation occurring. We don't want any summation happening that would lead to tetany. And by having this long refractory period, doesn't matter how fast the stimuli occur, tetany will never happen. And of course, if we had tetany, then that would stop the heart's pumping action. So if we take a look at this graph, this shows a graph of the cardiac muscle cell action potential. And if you look over here in the graph on the left-hand side, you can see here's the cell or this cardiac muscle cells at their resting membrane potential down here around minus 90 millivolts, and then at time 0, then we get this rapid depolarization and up to almost about plus 30, and then we see some repolarization, and then we have this plateau, and then we see some more repolarization. This purple graph shows how much tension or how much contraction will develop during this time. We'll talk about that briefly, but not really important in what we're going to talk about right now. If we look at this graph over here, this is a really good representation of the permeability to the various ions at the various time periods. So if we look at the initial depolarization of the cardiac muscle cells, we can see that their permeability to sodium is very great and very rapid. And so the voltage-gated sodium channels are going to open really, really fast, and a whole lot of them, and then they're going to close very, very rapidly, and the permeability drops back down here. Now, as these depolarize, you can see right about here at the peak of sodium permeability that we have the L-type calcium channels opening, and the influx of calcium into the cell, causing this this plateau right here. Now, we also have to take a look at what's happening with our potassium permeability. So this little decrease in the graph right here is corresponding to this permeability to potassium. So we have a little bit of repolarization taking place right here, but then the cell becomes more impermeable to potassium because of the multiple kinds of potassium gates that we have acting on our cells. But we want to focus now on what's happening with the calcium. So this plateau right here is due to the inward flux of this permeability, inward flux of calcium, and then the fact that these gates close slowly, so that we have this long absolute refractory period from the time that we have initial depolarization until we get back down here at complete repolarization at resting membrane potential. And so any stimulus that's applied in this time period right here will not be responded to because the gates have not reset. So, so if we want to take a look at that, here's some description that goes along with it. So I'll just leave that there, or you can go into the PowerPoint. So one of the things that we want to remember about cardiac muscle contraction is in order for it to continue, we need to have oxygen. We saw in the skeletal muscle, or in the cardiac muscle diagram, that there's a lot of mitochondria, and the mitochondria not only are going to use glucose, but they can use fatty acids and lactic acid as their substrate to go through cellular respiration. So the heart is never going to run out of fuel. There's always going to be something available for the heart muscle cells to use. But if they are deprived of oxygen, then that's where it will want to go into anaerobic respiration. But we don't want that to happen. And so when we have ischemia and we don't have enough O2, then we're going to get a drop in pH that will impair our ATP production, and we can't pump the calcium back out of either into the sarcoplasmic reticulum or back out of the cell, and then our gap junctions won't work, and our cells become electrically isolated, and so you know, we talk about this, the heart muscle dying. It doesn't die. It just cannot send signals anymore because it's become isolated from the rest of the muscle tissue. Okay, if we take a look at then an action potential that happens during or to the autorhythmic cells, so the intrinsic conduction cells, SA node, AV node, Purkinje's, we can see this graph, and as you can see, part 1 is our pacemaker potential. This is where we have those funny sodium channels, those hyperpolarized channels that will only open when we're down here where it's negatively charged, and so we have the sodium channels, those funny channels opening up, while we have the closing of the potassium channels. And also, this is important, notice that the membrane potential is never a flat line. We don't have a long resting membrane potential. As soon as we get down to the levels that are going to open those funny channels, then they open back up again, and then that proceeds to threshold. When we get to threshold, then that's going to open the calcium channels, and they will allow that action potential to happen. And then, of course, we'll have repolarization, and we bring it back down again, and we start the thing all over again. And so when the intrinsic conduction cells, or let's just say the SA node, so when the SA node is firing, this is what's happening. And so we just get the same depolarization over and over again as we get back down here to this level that's going to open the funny sodium channels. Now, one of the questions that seemed to give some people some pause was, well, what's going to happen? And I guess it's in a different study guide. But what would happen if if epinephrine or norepinephrine were applied? Well, it's just going to make these things more permeable to sodium. And so what that'll do is increase the slope. And by increasing the slope, then you can fit, so it gets to threshold much more rapidly, and then you can get more action potentials per unit time. And that's basically what's happening when we speed up the SA node. Conversely, when we slow down, when we're using the muscarinic, the cholinergic muscarinic receptors, because of acetylcholine release from the vagus nerve, then that will increase potassium permeability and slow it down, making this graph again lessen the slope so that you don't get to threshold as rapidly and you don't have as many action potentials per minute time or per unit time. Okay, so I think that's all that we need to take a look at for the 1.2 study guide on cardiac cell action potential. And this is a very important concept, these two different action potentials that we see in cardiac muscle tissue. So please make sure that you know what channels are doing what at what times in order to achieve the functions within the heart.